Hi, good afternoon. My name's Stephen Baker, and this is our laboratory at the University of Cambridge, where we run a research program trying to understand a little bit about how different microbes work, how they develop resistance to antibiotics and what we can do about them. But for the purposes of today, to carry on from something we've done a little bit before, and you may have seen us before, we're going to talk about poo. And for the purpose of that, this first joke coming up, this is my number 2 for today. This is Caroline. She's a PhD student in my group. Caroline, would you like to introduce yourself and tell us what you do and what you've done in the past?
Yeah, so I'm Caroline. I work with Steve and the rest of the group studying antimicrobial resistance, specifically in a type of bacteria called non-typhoidal salmonella. But before coming here, I worked at both the NIH in the US and also at a nonprofit stool bank, which we'll get into a little bit more about what exactly that means. But basically what I was working on is how we can modulate, change, study the microbiome, which is the community of microorganisms in and on us, to try and prevent disease and treat disease and better understand disease.
Okay, so we've got a few things here noted down that we're going to go through in the last half an hour that we're going to have a conversation about, and then we'll save some time at the end for any of you that may have any burning questions. So the focus of the subject today is we're going to talk about and give you a little bit of facts about poo. Okay, so first of all, some more comedic things and perhaps less scientific. That on average, I guess depending on who you are, we spend 3 hours and 9 minutes per week on the toilet. So actually, depending on your hourly rate, if you're at work, you can also calculate how much you get paid to sit on the toilet when you're at work. FMTs are a real thing. Do you want to talk about that?
Yeah. So
what's FMT stand for, firstly?
Fecal microbiota transplant. So basically, these have been sort of known in history for a really long time, since a guy in 500 AD in China, Ji Hong, he was the first person who ever described something called yellow soup, which was the first kind of FMT in history written down. But basically, it's the idea that some of us have what has been identified as a healthy microbiome, and that can mean many different things depending on where you are globally. And we'll get into more about some of like the paucities of data relating to microbiome health. But it's the idea that you can take someone who has a healthy microbiome, and then there's another person who has a dysbiotic microbiome or has an illness that's microbiome mediated or derived from a pathogen. And you can take that healthy person's microbiome and actually give it to the person who has that disease state and actually reverse the disease. So what it's become most commonly known for is actually a pathogen called C. difficile, which is really common in US hospitals, not as prevalent in the UK. But it causes a lot of morbidity and mortality, and increasingly it's really resistant to all available antimicrobials. So FMTs have become sort of this golden ticket where you can take someone's healthy stool and give it to someone who has C. diff and actually reverse their disease state within 24 hours. It's really amazing.
So you can actually eat someone's poo to make you more healthy, amazingly.
Yeah, there are crapses.
Yeah, so we as humans, anywhere between going to the toilet for a poo 3 times a day or once every 2 to 3 days is perfectly normal. Any less than that, then you have constipation and you probably need to go and seek some advice. And any more than that is usually associated with some degree of dysbiosis in the gut. And a very typical example is diarrhea. So if well, we've all had diarrhea, unfortunately, maybe some of us more than others. So diarrhea is then when we excrete lots of fluids from our gut. So we have an insult on our gut, usually a bacteria or a virus or a parasite. We respond to it and we try and excrete it, and actually we push it out of the body and we produce lots of water. Now, an amazing fact is pandas, not that I know a lot about diarrhea in pandas, but pandas can have up to 40 poos a day. So if you imagine that, so if they have diarrhea, then they're in big trouble. So what's in a poo? So what's in a poo, Caroline? You tell me what's in a poo.
Well, poo's mostly water to begin with.
Yep, 75% water, and the rest of it is not water. And I've got my notes here, so you can't see what they are, but I can remember. So it's 25% solid, of which 50% of that is bacteria, of which some are dead and some are alive. And that composites what we call, as Caroline mentioned, the gut microbiome. And then 50% of human cells. So 50% of that volume is actually us. So it's stuff that we excrete out of our gut that we get rid of too. but also it can contain fiber. So if you eat a high fiber diet, make sure you eat a high fiber diet because it's good for you. That is also included because you can't digest it properly. But also, we've heard a lot about microplastics in the press recently. So we ingest these from things that we eat also. So there's microplastics in our poo, and we're not quite sure what they do. But also precious metals. So there's a very, very small composition of things like silver and gold in things that we ingest, and they will also naturally pass through us. So if you want to be rich, then collect your poo and see if you can extract the gold from it. Right. Why is it the color that it is, Caroline?
Well, Steve, that's because...
refer to your notes, please.
It's because of the dead red blood cells and bile, which are awesome.
Yeah, so we have a lot of dead red blood cells. So we produce red blood cells, and when they die, we pass them through our liver and into the bile duct and that's what gives poo its traditional color. And it also smells really bad. Well, it should smell really bad, and it's because of a cadaverine, which is made up of a chemical called stactol, stacatol, which is a tryptophan derivative, but also can change depending on the chemicals that you eat and the types of food that you digest. So if you eat particular types of food, I'm trying to think of an example. Onions,
red beets. So you can't red beets.
Oh, red beets. Sorry,
so you can't be a fecal transplant donor if you eat red beets,
right? Because
it makes it look like you have blood in your stool.
Right. Okay.
So, yeah.
So what we talk about then and what we work on here in this laboratory is very much focused around bacteria. So what is a bacteria? A bacteria is a single cellular life form that can replicate itself. So unlike us, where humans reproduce sexually, these things reproduce asexually, and they live everywhere, and they're very, very small individual cellular life forms. They're amongst the most simplistic cellular life forms, single cellular life forms there are all. And they're everywhere. So they're inside us, in our gut, as I mentioned to you, inside our mouth and other parts of us. They're on our skin, they're in our hair, they're all over us. And actually, depending on how you look at it, and if we count up the number of cells in our body and the number of bacteria we have that live in us and on us, actually there's 10 times more bacterial cells than there are human cells in us. So depending on how you want to measure it, actually we're 10% or whatever it is, 100% more, 10 times more bacterial cells than we are human cells.
Yeah.
And they perform amazing roles. So we think traditionally of bacteria as being very bad for us. That was when they were first discovered and described, they were associated with the organisms that cause disease. So we assume that they're all bad for us, but actually then they're not bad for us. And some of them have very, very good functions from us. And in fact, we can't live without them. So do you want to give an example of some of the things they do?
Yeah, they're pretty amazing. I mean, they can, we're talking about breast milk, things like that, right? You know, lactobacillus, fiber fermentation, you know, colonization resistance against other pathogens.
Yeah, we can do all kinds of things with them as well. We can manipulate them in different ways, and we get them produce methane. They're very important in food production, but also they have a really important beneficial impact on our health. So they're not all bad. They're the most abundant life forms on the planet. So those of you that are working virology will go, no, viruses are. Viruses are kind of slightly different thing, which we can talk about later or someone else can talk about. But actually bacteria are the most abundant single cellular life forms on Earth. There's 5 trillion of them on Earth, and they're everywhere in every environment that we've ever found. Now, this is an amazing statistic. Again, this isn't a particular focus of our research. This is due to the internet, but if you lay all of the bacteria around Earth end to end, which would be tricky to do, but you know, we could give it a go, they would be 10 billion light years long. So that's how many there are on Earth. If we line them all up on Earth, they'd be 10 billion light years long. I don't know why you want to do that, but if you, after a project, we could start now and see how far we get.
The next Tesla.
Yeah, they've also been around for some time. So they're estimated to be 3.5 billion years old. And we don't know really their kind of defined origins of how they originated, but probably from the early life forms of the ability to then produce nucleic acid into amino acids and form single cellular structures. Now, when they first arose then, the world was a very different place, that the world had no or very little oxygen, and they were all anaerobic. So when we started getting plants on the planet and they started to produce oxygen, these bacteria had to adapt and then start to survive. In fact, oxygen was highly toxic for most of the bacteria on the planet, but some maintain their ability to survive without oxygen. Our gut is a very low or no oxygen area, and this is what we call anaerobic environments. So actually that is at the gut, the organisms that live in our gut that are anaerobic actually hark back to the early days of the planet when they originated, when there was no oxygenation. So we have lots of different types and different ways we can categorize them, but we have anaerobic bacteria, which can survive without any oxygen at all. And we have aerobic bacteria, of which no oxygen is actually toxic to them. Interestingly, most bacteria, even though we know a lot about them, we work on them, when we can grow them, when we're getting better at growing different types all the time, most bacteria we think are yet to be identified and categorized. So we have no idea really about the bacterial populations that live in the deep oceans. We have no idea really about the big bacterial populations that may be surviving in parts of the Arctic or the Antarctic. These places have always been sampled, but only in very small amounts, so we don't really understand the magnitude of them. They're very fast, so amazingly we don't associate bacteria with being quick, but they can move 25 times their length in 1 second. So if that was you, then you'd be running the 100 meters incredibly quickly. And they're very, very good at reproducing. So E. coli can reproduce every 20 minutes or so, which means after 7 hours, one bacterial cell can produce about 2 million offspring, which makes them incredibly prevalent and incredibly good at surviving.
And that's part of why you can get sick so quickly, right?
Yeah, so if you get sick so quickly, because if you then have a small amount of one, they can replicate in your gut, and then suddenly you have a big population of the organism in your gut, and that's what makes you sick. Now, we haven't, as yet, of today, of going to air, found life on other planets. If it happens now, then I do apologize, but we haven't. But I would predict that this may happen in coming years, hopefully within my lifetime. And I predict that if we do find life on other planets, then there's a very good chance there's going to be evidence of either living or fossilized bacterial cells. So they're everywhere. They're really abundant on the planet, and there's a very good chance if we have similar environments on other planets that actually we might find bacteria on other planets as well. Okay, so let's talk about microbiomes. Okay, so we mentioned biomass of bacteria. Okay, so there's about 70 gigatons of bacteria on Earth. That's estimated. I don't think anyone's weighed them, but that's what's estimated. 70 gigatons of bacteria.
What's a gigaton?
A gigaton is 1 billion tons. So that's 70 billion tons of bacteria on Earth. Let's put that into context. The biomass of humans on Earth is 0.06 gigatons. So if you can work that out, I can't work out the magnitude, but actually the bacteria are far more abundant than humans. Now, a microbiome is the composition of all bacteria that are in a certain environment. Isn't that correct? So you're the microbiome expert, so tell us a bit more about microbiomes.
Well, so microbiomes, they develop in utero. So there's been a number of studies that have shown, depending on the way that you were born, cesarean section or vaginally, your microbiome is going to look a little bit different. Depending on where you grew up, it's going to look a little bit different, depending on whether or not you're exposed to antimicrobials chronically, and depending on what you eat as well. So it also does change as you get older.
Yeah, so it can be impacted by lots of different things. So it can be impacted by your diet, by your age, the environment that you're in, and also your health state. But one of the key things, or arguably the biggest thing for your microbiome adapting, is when you take an antibiotic or an antimicrobial. So antimicrobials work non-selectively, which means when we take one, they'll make you better, hopefully, by killing the organism that we think is causing the infection, but also will kill everything else. So they have a fairly big impact on changing the structure of your microbiome, and actually can often, depending on the infection, make you feel worse from the composition of the microbiome.
Yeah, it sometimes has been described as like killing an entire forest, because you have so many different species present in your microbiome, but then you're actually sometimes depleting the entire forest.
Yeah, so the gut is probably not only the most complex microbiome that we have, so we have them on our skin, other places in our mouths, but the gut is the most complex because of the scale of our gut. The gut is actually, if we think about it, an external surface that's inside our bodies. It's a tube that goes all the way through us where we digest food, and the bacteria are really key. And we've studied this to try and understand how it impacts on different things. And we've come up with this concept of what constitutes a healthy microbiome. So what constitutes a healthy microbiome?
Well, it really depends who you ask. And it's also interesting because we really have a lack of data outside of North America and Europe defining really what a person's microbiome looks like at all. And we have a lack of longitudinal microbiome sampling as well. So, you know, I think a healthy microbiome, it depends who you ask, but I would say a lesser prevalence of antimicrobial resistance genes generally. And
yeah, so we think that a healthier microbiome is associated with diversity. So we think that the more different types of organisms you have living together in this community in our gut is healthier for you. And lots of things can affect that. And we're just learning now how the different diversity in the microbiome can be moved in different ways, can impact not only your gut health, but also your physical health, your response to other things, your inflammation, and also now we're considering even the impact on mental health too, that we think that manipulating or understanding certain aspects of your gut microbiome may have a really important impact on mental health. So it's really a key area for research, but also a really key area for pharmaceutical companies to try and develop new interventions of how we can control or manipulate aspects of it to improve health.
Yeah.
So as you said, there's a bit of a focus on microbiomes from people from the US and Europe.
Yeah.
So do you want to comment on why that is and what we can do about that?
Yeah, I mean, I think it sort of originates because a lot of the investment from funders or governments has been looking at diseases that are prevalent in Europe and North America. So, you know, governments investing in things like cancer and inflammatory bowel disease, which are really prevalent and increasingly growing in these populations. But just because governments are investing in their own countries on these things doesn't necessarily mean that the microbiome isn't implicated in diseases in other places. So there have been really interesting studies showing that microbiome is actually tied to malnutrition, and that children who are malnourished and who have recalcitrant malnutrition actually have different microbiomes than children who recover from malnutrition or children who never have malnutrition at all. So yeah, I think.
Yeah, there's a huge bias in the way we've conducted science into microbiome research, and hopefully that will change in coming years. But we don't really understand the implications of what's going on in particularly in developing countries, and how the impact of maybe different diets or malnutrition or other comorbidities, other diseases can impact on the microbiome, and what that means for healthcare within those places, but also means for long-term health inputs for children, including the way they respond to future infections, including the way they respond to immunization, and all of these things are not yet clear. So there's a huge amount of work that's been done, but it's been very focused in the Northern Hemisphere, and actually by shifting our focus perhaps to developing country, we'll learn more, but also then how we can then provide better healthcare for people that are less financially secure than we are here in the West.
But I mean, these are this totally new class of drug, right? I mean, these microbiome-derived therapeutics. So, you know, regulatory authorities in the Northern Hemisphere getting used to what these look like. Hopefully, then that will pave the way as well for it to be easier.
Yeah, and maybe some of you take a probiotic or eat probiotic kind of yogurts. The way those do or don't work, again, we don't really understand. There's this concept again that's been promoted for these things, that they're good bacteria. They're certainly not detrimental, but we don't understand enough about those specific organisms to know what the benefit is for your health. We do know potentially that certain types of bacteria that are found in probiotics may have a positive effect, but actually even that, we're not yet good enough to really understand how specific probiotics have a particular input into a certain disease state. So probiotics are probably good, but the way we use them at the moment is probably not really optimum.
That's interesting too, because I mean, in some of the preliminary evaluation of microbiomes that's been done, things like Prevotella have been found, species bacteria has been found to be more common outside of Europe and North America, whereas Bacteroides and other species have been more commonly found in more industrialized countries.
Yeah, so we're really trying to get to grips with this and understand not only how this can change, how it can be impacted, but also what's going on in developing countries. Now, something that I want to make sure that we talk about while we're here is then something that we mentioned, which is probably a key kind of subject of people have thought about, is antimicrobial resistance and what that means. And it's a nice kind of movement on from the microbiome, because some of our work here has really focused on understanding the composition of the microbiome, particularly in children in developing countries, but also the huge amount of antimicrobial resistance genes we find in these children. So traditionally, I think we think about drug resistance as being a problem for when you're infected with a bacteria and then you don't recover from the specific antibiotic. And that's completely true. But what we're starting to understand now is this is a really kind of normal state, that it's very common, incredibly common for children and also adults, but mainly children in low and middle income countries to carry a whole host of different resistance genes in their microbiome. That means that those resistance genes can then be transferred to other things and they become a major reservoir for how these things move around. And that's kind of the work we try and investigate. So a pathogen is an organism. In this case, we're talking about bacteria. So let's say a bacteria, but it can be other things that causes a particular disease. So we're talking about the gut. So we ingest a particular type of bacteria. So when you get diarrhea, unfortunately, the bad news, I'm telling you that if you have a bacteria that's caused diarrhea, you have eaten that, which means that actually that material you've eaten has got some fecal material on it with the bacterium which has made you sick. So every time you get diarrhea, unfortunately, the bad news is you've eaten something, either an animal or human poo, which has made you sick, unfortunately. That's the truth. So we try and learn a little bit about that too, but that's a pathogen. And actually, ordinarily, most of us will recover from these infections, hopefully, without the requirement for an antibiotic.
Hopefully.
An antibiotic is originally a naturally occurring chemical that there's a large history behind, and then they've been developed over the years into different chemical classes. And what they do is they aim to either stop the bacteria replicating or kill them outright, which then makes us recover. Now, unfortunately, which is largely a natural process, but it's been driven by mankind and our use of antimicrobials, that these organisms are starting to become resistant to these antibiotics, which means that when we take them, it means that the drugs don't work as effectively as they used to, or sometimes they don't work at all. Now, this is then key because there are lots of different types of antibiotics. So the deal is we take one, it doesn't work, we just take another one because there's lots of alternatives. That's kind of true, but what happens is the bacteria acquire resistance to lots and lots of different antibiotics. So one definition is something called multi-drug resistance, where we say an organism is resistant to 3 or more classes of antibiotics. And then there's another one, which is XDR, which is extended drug resistance, whereby the organisms are resistant to all but 1 or 2 limited class of antibiotics.
Yeah.
Now, I realize, Caroline, I've just done quite a lot of talking. Would you like to say something about drug resistance?
I was just going to add that there's also pan-resistant organisms.
Oh yeah, pan-resistant. Yeah.
Which basically, and correct me if I'm wrong here, but that basically means that pathogen isn't susceptible to any.
Yeah. So we're yet to see this particularly commonly in the UK, but I spent a lot of time working in Asia, and certainly we would have cases in the hospital I was working with where we'd see organisms that were pan-resistant. So these were children, adults infected with severe infections, sometimes in the chest, sometimes in the blood, and these organisms would not be susceptible to any antibiotic that we could access, which means that actually they had no treatment. And not that I want to scare people, but this is a real phenomena that's not something that we need to think about that may happen to us. This is happening to us now. And this is why we do work in these type of laboratories to try and understand this process, but also try and work out what we can do about it to try and prevent or treat these infections as they arise. So again, not to scaremonger, but just some kind of facts. The impact of drug resistance on healthcare is likely to be $5 billion per year. So that cost comes from having to treat people with different antimicrobials. People with drug-resistant infections stay in hospital longer. They require more intensive treatment. They often have worse outcomes, which means they do worse, which means they require more medication. And actually, they're likely to cause more deaths than cancer by 2050. So whether we like it or not, and the pandemic has really kind of personified the way we think about the threats now from infectious diseases. And the pandemic has been devastating in so many ways, from a healthcare challenge, from people losing family members, the impact on the healthcare system and financially. But AMR has an impact that could be much greater than the ongoing pandemic, which is why we need to start thinking about how we treat it now. Okay, so and where do we use them, Caroline? Okay, so where do we use antibiotics? Do we just use antibiotics to treat infections or not?
No, they're also used in agriculture quite commonly to prevent infection in animals, to fatten animals, and also sometimes prophylactically as well in healthcare. Not
Yeah,
just treatment.
so again, unfortunately, we think of antibiotics as these kind of magic drugs, but we're not very good at protecting our magic drugs, and we overuse them. Overuse them is a kind of complicated term. Their use is highly restricted in the UK. In other places, it's less restricted internationally. And if you were sick or even not sick in Asia, in many places, you could go into a pharmacy shop and just buy antibiotics and take them yourself. And also, they're often given to animals to supplement growth or to stave off infection, not necessarily without any requirements. And do we find them? Where can we find antibiotics then?
Well, we find them in wastewater quite often.
Yeah, so we can find large quantities of antibiotics. So we take them, we ingest them, we excrete them either through our urine or through our poo, and they end up in the environment as these small chemical molecules that we can identify, and we can find them in wastewater, we can find them in river sediments, we can find them in fields, we can find them pretty much anywhere. So we can find these antibiotics. But then the bacteria have to survive in these places. So what also do we find in these places if we find drugs? What else do we find?
Bacteria?
You're looking at me, yeah, we find drug-resistant bacteria. So because this is not just necessarily a thing for us, that we push these things out everywhere, that we find drug-resistant bacteria pretty much everywhere we look. So we found drug-resistant bacteria in almost every location that's ever been sampled. Some of it dating back historically to the pre-antibiotic use, when the genes that made them resistant naturally occurred but did something different and they've adapted to antibiotics. But also now, if we sample them in the West or in Asia, or in Africa, then we can find them pretty much everywhere.
Yeah.
Okay, so drug resistance then, what can we do about it?
Well,
I was asking myself that kind of thing, but you feel free to answer.
I was saying we can be better stewards of what we have.
Yeah, we can use them better than what we have. It's a natural phenomenon. It's not something we can just stop, or we may be able to slow it down. We probably can't reverse it. But this is the way bacteria evolve. So we impose a selective stress on them, and then they come up with mechanisms either by acquiring resistance genes or going through a process of mutation whereby they change their genes, which allows them to survive. And this is a perfectly natural process. But what we have to do then, is have better control over the way we use antibiotics. For you people out there, perhaps questioning whether you need antibiotics, certainly never demanding that you're given antibiotics if your doctor doesn't think you need so, but also recommending the way they're supposed to be used, that we shouldn't be giving them to other people. We should probably, although this is maybe controversial, complete the course that's given on the thing. That's what we always say. I don't think there's any good evidence to support that, but we should probably use the way they're marketed to be used. And then we should be really sensible and thinking about the way we approach this and try and look after our microbiome as well, because it's not just us getting better, but also the impact on our guts.
Yeah. And what about at a policy level?
Yeah, at a policy level, at an international policy level, we really need to review how we provide access that we need. The world has a big human population. We need enough food to survive, both vegetables and also animals. So the supplementation of antibiotics has actually helped sustain the population growth. Now, there is data to suggest that actually if you remove antibiotics, there is a drop in productivity, but then it rebounds after 3 to 4 years of cycle. So actually, we could then start to question whether we really need to start using the amount of antibiotics we use in agriculture. And also then in developing countries, we should probably we should certainly act upon antimicrobial access and how we really preserve these things as they come along. Okay, so we've got a few minutes left. So something then I just do want to talk about then, two things before we go on to some questions. So if we're so good at the work that we do here, not particularly me, but in science, then we should be able to come up with new ways to kill organisms. Yeah. So, but there is an issue that we've been really good at finding new antibiotic classes which kill the bacteria in different ways. And fundamentally, there's only certain ways we can kill them, because what we don't want to do is take chemicals that harm ourselves. Antibiotics work by stopping a process that only occurs in bacterial cells. And we're getting to the end of that, probably that resource of knowing how to target. There are some probably still left, but they're getting smaller and smaller. So the process of developing new antibiotics is costly because there's a lot of research needed. It's very difficult because we've been through a lot of these processes before, and actually we're starting to get the end of those resources. But also then the market for them, whereas we think is big, is actually relatively small. So pharmaceutical industry don't make a huge amount of money from antibiotics because they're so cheap. So when we do develop these new drugs, we need to think about how we use them and how to preserve them, but also make sure there's an interest from pharmaceutical companies to develop.
Yeah, which can require government investment, right? Because the way most infections work is that you give an antibiotic and then you no longer have the infection, which, unlike a disease that's lifelong, or something like cancer that could come back, most infections happen just wrong.
Yeah, most infections either resolve or they have a bad output, and therefore the duration you take the antibiotic is relatively short, which means that pharmaceutical companies don't make a great amount of money for them. So there needs to be subsidies if we do develop new drugs about how we use them, but also how they're developed, to make sure that actually we're in the process to take them forward. Yeah. And just as the last kind of comment then, so part of our work and the way we think about these things is, we think about and coming up with new processes. So we're not just dedicated on developing antibiotics to kill bacteria. There are other processes. We think about how to use different chemicals. We think about how to use our own immune system. So how can we manipulate our immune system by activating different components of it to better kill bacteria? And we have some work on that. But also perhaps even more aptly at the moment, given the impact of the pandemic, is develop vaccines as well. So thinking about how we can actually target our immune system or program our immune system to actually kill these things before they can cause an infection, which means actually we never then require downstream antibiotics, because hopefully our immune system will work effectively. So we've gone through half an hour there of a fairly kind of whistle-stop accelerated tour of bacteria, of microbiomes, of how we can manipulate the microbiome, mentioned diarrhea. We've mentioned some different comical and perhaps not so comical aspects of poo, but also then AMR, how antibiotics work and what we can do to maybe slow that trend down and also develop new things. I would say that actually then, apart from it's my own personal view, because this is what I kind of get paid to do, that I think that understanding the way these things work and understanding how we can prevent and treat drug-resistant infections is probably a bigger challenge as climate change, and something that humanity really needs to then not just think about, but really start to take seriously. And we need to think about how we can protect ourselves better against the infectious disease threats of the next 40, 50, 100, 200 years.
I would say even sooner than that.
But, well, ultimately, yes, but we need to do it now, is my point. Okay, so I think that was us. And I think now we have some questions that we'd like to go through that we've got here on the tablet.
I just want to say that we're not clinicians.
No, I'm not a clinician. No.
Okay, so one person has asked, after taking a course of antibiotics, does your gut microbiome eventually return to its original state?
Oh, wow, what a fantastic question. So, so largely, yes. So it will. So what happens is we wipe out large aspects of it in one go when we take it. So as Caroline alluded to, taking an antibiotic is largely like an atomic bomb going off in your gut. That you would kill everything that antibiotic is susceptible to that antibiotic, and it will recover, but it probably won't recover completely. That there is some evidence suggests that we get like a microbiome scar, a signature that actually that it will have been adapted with time, and it will recover to the point where we have a multitude of different organisms, but there will be a signature there that you've received an antibiotic, and we don't know what the long-term detrimental effect of that is. But yes, if you can identify people that received antibiotics a long time after they've taken them.
And that can be sometimes why, if you have multiple insults, then being on multiple antibiotic courses can end up
Yeah,
having
so
this
multiple
kind of outcomes.
antibiotic courses can have really detrimental effects, and
Yeah.
this is why we often sometimes acquire these C. difficile infections.
Yeah. Are intravenous and oral antibiotics equally likely to wreck the microbiome?
Oh, wow. Another great question. So yes and no. So it depends very much on the class of antibiotic and not the route of administration. So some drugs will have their focal activity in the gut. So taking an oral antimicrobial, we absorb it through the gut, and that would mean then that there will be some activity within the gut surface which is likely to kill the microbiome. That's true. But there may be also different antibiotics that may not be particularly well activated inside the gut, but activated inside the blood that we take orally. So it depends very much on the pharmacodynamics of the drug that we take, and also its excretion route. So some drugs accumulate in the gut and will be excreted through the gut, even if we give them intravenously, and other antimicrobials may be excreted through your breath, or either through your skin, or either through your urine, depending on the class. And some will have a more detrimental effect than others.
Someone has asked what we can eat to improve our gut microbiome outside of fiber. Any other recommendations?
Yeah, so I mean, so we've talked about fecal replacement therapy. I wouldn't suggest that you go down the local farmyard and start picking up cow fats and eating those. Please don't do that. But having enough variation in your diet. So and kids, you know, are the world's worst for this, of eating pretty restricted food. So eating as many green things as you possibly can, and having a balanced diet, all the tedious things that you're told at school, to avoid sugars, to avoid saturated fats, and all these things, they have a really detrimental effect on your microbiome. And we know that people that go on and develop certain metabolic diseases, such as obesity, by eating sometimes not varied enough food with high amounts of fat and sugar, have very restricted microbiomes. So we think that by having a varied diet and eating, unfortunately, as much healthy food as you can, probably has a better effect on your microbiome. And also dairy products as well, milk, yogurts, and these things are good for our microbiome.
Fermented foods.
Fermented foods, yeah.
What use could be made of phage for combating bacteria?
Ah, wow. Okay. Yeah, so phage, a bacteriophage is a virus that only infects bacterial cells. It won't infect our cells. And phage are very good. We can populate them. We do grow them here. And actually, in a laboratory, they're very effective at killing large amounts of bacterial cells. And there are now coming to market some phage therapies that we think have some particular activity against certain types of bacteria. And there's been a lot of work historically done of this in parts of Eastern Europe. And I believe that phage therapy is still a big thing in parts of Eastern Europe. Less common here, but it's coming on board. It's an area that we need to explore. It's not that effective for particular types of infections, particularly those in your gut, and we don't really understand why. But certainly for some topical infections, then it can work very well. It's just not a particularly kind of as developed science as perhaps some of the other treatments that we take.
Yeah, and I think from a regulatory perspective as well, there's going to need to be a lot of work there.
But huge potential.
Yeah. So we have a couple of questions about children eating soil and the West being obsessed with hygiene.
Yeah.
So the hygiene hypothesis wasn't really something that we touched on.
Yeah.
But
yeah, so the hygiene hypothesis is that as we've developed these kind of bacterial-free environments in the West, that our immune systems have become weak, and we've started to develop these syndromes such as allergies quite commonly. There is some truth in this. So we know that exposure to a wide variety of things can probably have a positive effect on the way your immune system develops. And we know also that the more kind of common allergies that we may see here are less common in developing countries, and particularly probably less common in places where children may be drinking contaminated water. Now, there's two arguments here. First of all, we're potentially too clean here with the way we work, but also we know that drinking poo in contaminated water is not good for you. So there's a balance here somewhere about the way we kind of trigger our immune responses to make sure that we're not susceptible to either other infections or autoimmune diseases or allergies, but also ensuring that we're not exposed to so much that we then have other detrimental effects from giving ourselves too many pathogens. So yes, this is completely true, that probably we are too clean in the way we think about bacteria. And I'm not suggesting again that we go out and eat a cow pat, but we need to then probably not be so paranoid about the way we expose everything to bleach and try and kill everything in our houses, because some bacteria, as we pointed out for your microbiome, are good for you, and the more variety they have, the better.
Yeah, a couple of people have asked how FMTs or fecal transplants are conducted, and it depends what your disease state is. So you can either take what are called poop pills, or crapsules, as I referred to earlier.
Crapsules, did you say?
Crapsules, yeah. Or you can have an enema or colonoscopy delivered, or even a nasogastric tube. So obviously, I think most of the companies that are trying to develop these
all sounds gross.
these therapeutics are aiming to have a small number of capsules that you can just take orally, similar to how you would take antibiotics. I can't really envision it ever being intravenous.
Have you ever taken one?
No.
Okay.
A couple of people have asked about microbiome changing with age, and if it can be associated with certain disease states as you get older.
Yeah, it does change as we get older. It becomes less complex as we get older, probably because as we essentially become less complex, our cells start to wear out a little bit, and therefore we have a little bit less dexterity all over our bodies. So yeah, it becomes probably more fixed and becomes less diverse. We don't really know what that means, but there are investigations going on, and we know that some aspects and some organisms in the microbiome can trigger some detrimental effects, such as colorectal cancer and other things, of which, again, it is in its infancy, but we do believe that certain aspects of the microbiome, as we hit certain types of age, may have a detrimental effect, and we don't really understand why.
A couple of people have asked about what we can do, or it's okay. This question I'm looking at specifically, can you say something about how disease pathogens might modify the gut microbiome?
Yeah, so they might modify the gut microbiome in many different ways. So they can actually kill other organisms that are resident within the local gut. So they may actually, to reduce the amount of competition, they may actually then release a chemical which can kill other organisms in the gut and therefore deplete that aspect of it. But also then they induce inflammation. So some pathogens, as they attach to the gut surface, the gut becomes inflamed. We produce an inflammatory response, an immune response, and that can also then have an impact on changing the structure of the microbiome. But also then just by physically the amount of fluid that we need to actually remove from our body. So a response to us having these pathogens, something triggered by whatever organism is, that as we produce lots of fluid in our gut from the inflammation, or as a response to get the bacteria, the virus, or the parasite out the gut, it can also then wash out and deplete the microbiome. And what we've seen in work that we've done actually is then it recovers by being colonized initially from organisms that are in your mouth. So at the early stages of diarrhea, they get wiped out by the diarrhea, they leave a gap, and then as we swallow, organisms from the mouth actually then begin to colonize and reconstitute your gastrointestinal tract.
It's really interesting. How much work is there on novel antibiotics or antimicrobials, not just antibiotics?
Yeah, not as much as there should be. So basic science, such as conducted in universities, there's still quite a lot of interest in this area. Kind of almost the philosopher's stone for many bacteriologists, including myself, is working out how we can kill bacteria, because we know that's good for healthcare. It really falls by the wayside as we try and progress these things. Us doing experiments and working out how to kill bacteria with particular chemicals in a laboratory is one thing, but then how do we turn those into products with pharmaceutical companies becomes another one. And actually, the discovery pipeline and the development pipeline for antibiotics in pharmaceutical companies is almost completely dried up. So there's a lot of basic research ongoing and working out how we can kill bacteria, but actually very little of that is picked up and then translated into kind of real medicines.
And when you're going about trying to develop new antimicrobials or treatments for these different pathogens, are you people have asked, are you trying to develop an antimicrobial that works against just a specific pathogen, or are these more broad spectrum?
Yeah, so we do a little bit of both, and there's different approaches. Probably pharmaceutically, the best working drug would be one that has a broad spectrum approach. What we focus on here is trying to kill very specific organisms. So we try and find certain markers or signatures or tags on the surface of the bacteria that then we can then hit with particular approaches. So yeah, so I think that probably most of the focus research is still on broad spectrum, and that's where the interest is. But actually, I think we're starting to accept now that we need to start focusing on trying to identify narrow spectrum targets.
And are there some unique ways that we're using technologies in this lab to try and identify those tags? And
yeah, so we can do all kinds of different things. We have different approaches by taking libraries of organisms that we make specific mutations in them and hitting them with different chemicals, or hitting them with different cocktails of antibodies, and then trying to work out which ones survive and which ones die, and then trying to work out from the genetic information, what that correlates with a particular protein or a target on the bacterial surface.
Wow, you guys are asking a lot of really interesting questions. Okay, let's see. Someone has asked what other things are present in our gut. So fungi.
Yeah, fungi, viruses, plant DNA. So the microbiome, we often think about then taking the biomass of the gut and then throwing it, if you like, throwing it's a bit of an unsophisticated word, performing a complicated biological process to sequence it. But that also means that we don't just sequence bacteria, we sequence everything else there. So there is non-living things, as I said, or there's microplastics, bits of fiber, but also if we were just to look at the DNA content, there will be human DNA, there will be bacterial DNA, there'll be viral RNA, there'll be viral DNA, there'll be fungi, and there'll be quite a lot of plant material in there as well. So lots and lots of different things.
But I think relative to the amount that we've studied the microbiome, if we're considering the microbiome just as bacteria, the fungome has not really been studied at all.
No, the fungome, if that's a word, if not, we've just made it up here. I think it probably is a word. The fungome is not something that's particularly well understood in the gut as yet, and how that changes. No, I agree. We're very much focused on the bacterial component of it because we think that's the most important aspect.
Yeah. A couple of you guys have asked about our view on prophylactic use of antibiotics in animal husbandry. I don't know if you have any thoughts.
I'm not a big fan. So there's various evidence out there to suggest, as I say, that it should be a restricted policy. We don't do it in the UK anymore. So tetracycline was used for many years as a growth promoter, and I think that's been banned across the European Union, of which we were a component of at one point, but now we're not. I think those rules and regulations are going to stay. But it's very common in across Africa and Asia, and it's probably something that we need to reduce. One of the big kind of hypotheses is that drug resistance in animals, because we use so many antibiotics in animals, are spread to humans. To me, that's not particularly conclusive. My personal opinion is that we shouldn't be using large quantities of antibiotics because of the way they may impact on long-term health for humanity, but also the planet and the environmental contamination. But my personal belief is that when we acquire drug resistance, that drug resistance bacteria are acquired from person to person, rather than us acquiring it directly from animals. Is some data that we've generated that I think is probably more realistic. And
so, slightly more basic question, or fundamental question, is how long do bacteria live?
How long do bacteria live? That's very interesting. Yeah. So bacteria, depending on the bacteria, some of them can live only a few minutes, depending on how we cultivate them. In a laboratory here, we can keep them alive, and we can kill them, and we can manipulate them for as long as we want, really. In our gut, because we excrete them, we appraise them fairly regularly, so it's really difficult to measure. But also we know that we've recovered bacteria that have been in ice cores. We can do something called lyophilization here, or freezing, which means that we can take them, we can actually dry them and then keep them indefinitely. So the reason I said earlier about if we're going to find life on other planets in my lifetime, it's like to bacteria, is because bacteria can survive long-term in all kinds of conditions, on us, on surfaces, in water, anything. So actually they can survive for millions of years.
This is a really interesting question. Why are fecal bacteria harmful when we ingest them, but why are they happy when they're just living down in our guts?
Yeah, okay. So it's because it's the composition of what's in them. So they're happy living in our gut because our gut is a partitioned environment where they've learned to replicate and survive perfectly happily, and that's their place. They're comfortable in that environment. There's a it's
optimized for them.
It's an ecology whereby they're comfortable, and if we then somehow disrupt that environment by introducing bacteria, and remember that what's coming out of our bum may not be the same as what's in our upper duodenum, then we upset that environment by exposing those organisms to that. So meaning that actually just because something that's in our poo is in our gut, that isn't necessarily representative of what's going on in all of our gastrointestinal tract. We'll have different compositions of the microbiome all the way down our gut, which then if we then change that in any way with a drug or introducing some fecal material, for example, then we'll disrupt that very subtle change in the microbiome in different places in the gut.
Wow. Someone has asked what alternatives there might be to antibiotics to treat bacterial illnesses if they become increasingly resistant.
I mean, so this is again a controversial topic. At the moment, there isn't anything that's really obviously coming forward. There is no new technology that we think is going to be a forerunner. There are different approaches that we think. Someone mentioned bacteriophage. Something that I'm particularly interested in is antibodies. So how we can actually generate and manufacture antibodies that can have an effect whereby we trigger downstream immune responses in ourselves. And I think that probably is going to be the most likely view. The other thing is that people are developing specific antibacterial peptides. So these are small peptides, groups of amino acids that can actually recognize the organism and have a detrimental effect to the organism to stop it doing something. And these are fairly well researched, if you like. We understand a lot about them, but not at the point that we can really translate them into anything soon. But we are interested in these approaches.
Wow, great. Someone has asked about MRSA and why it's such a problem.
It's less of a problem now. So MRSA is methicillin-resistant Staphylococcus aureus. It used to be a big problem, particularly in healthcare facilities in the UK. It originated probably in the 1980s and 1990s and then spread everywhere. It's a big problem because it's resistant to the particular drug, so methicillin, that was commonly used to treat staphylococcal infections. And as it became resistant, then these conditions become more difficult to treat. And if they become more resistant, then you're required on more serious drugs like vancomycin to treat the infection. So the concern is that this is a kind of pathway for them becoming resistant to multiple antibiotics, and we have to then start using more complex and more expensive antibiotics.
So kind of tying into what we were talking about at the end, what do you think is the economic potential of poo?
The economic potential of
that
poo
could be in terms of the detrimental effects of poo as a pathogen, the components of pathogen, or
I mean, I think poo is the most fascinating substance there is on the planet. It contains so much information about an individual and environment, and I think that realistically we're only now just beginning to understand the potential of how not only we can manipulate our own, but how we can take aspects of that, be it particular chemicals, particular organisms, or particular chemicals produced by particular organisms, and really turning something that can have a positive effect on human health. And I think that, in 5, 10, 15 years, I think fecal replacement therapy, but also how we learn to manipulate the microbiome through the use of key probiotics is going to become really important to healthcare.
No, it'll be interesting too what kind of drug combinations we could do with some of these different technologies you're describing.
How are we doing for time?
Okay,
we've got 5 more minutes.
Wow, okay.
We've got five
Lots of questions. Okay. People are asking about CRISPR and how it could be used to target drug-resistant bacteria.
Yeah, okay. So CRISPR technology is a gene editing tool which allows you to target specific things and knock out specific genes. So the concept with kind of CRISPR technology is you can target particular pathogens and you can insert these particular markers into the chromosome of the pathogen and knock it out. So yes, and we can do that quite comfortably again in a laboratory. The technological problem which we then have to solve is then how we make this into a form of medication that allows us to take it and activate things on our bodies or in our bodies, and that becomes a problem. So there's lots of things that we can do in a laboratory that can kill organisms, such as CRISPR techniques, such as bacteriophage, but then the technical problem is how we turn them into a medication. And again, CRISPR technology is something that's really interesting, that may develop into something major, but at the moment we don't really have the know-how to turn it into a drug that can be manufactured and then used routinely like an antibiotic.
So a couple of people have asked what the easiest way is to assess the health of their own microbiome, or to get information on if they have a healthy microbiome.
Yeah, I'm surprised there's not companies doing it, but there's something.
Well, there are. Well, there are a couple, but they've gotten embroiled.
Oh, really?
Political, at least in the US.
Okay, right. So I mean, you can send your poo away to places and they will tell you what's in it. I don't know whether there's anybody doing that in the UK, but we did talk about it once, about having a kind of, you can generate your own signature of your own microbiome by sending away somewhere, which personally I think would be really cool. I mean, I don't know why, but it'd be really good to have like your human genetic information, having your,
at this moment in time.
Yeah, right, having a signature of your own microbiome. But yeah, I don't know whether everybody's doing that. Google it and have a look and see whether, if so, poo into a jar, put it in the post, make sure you do the lid up properly and send it off and see what you get back.
Make sure you put the right address.
Yeah, make sure you put the right address on you. But otherwise, then I think that we kind of know when we have a problem. You know, we know what our poo looks like. We know how it smells. We know when it smells different or smells really bad. We know when we have diarrhea. We know when it's a funny color. And those will be signatures of change. How we then go around and test things ourselves, I think that maybe this will become more common, where we will start actually having routine testing of your microbiome to see how that impacts on human health when we understand more about it. But at this point in time, apart from observing it, unless you can find someone that's going to do it for you on Google, there isn't really a good way.
So another question, kind of following on these things we've been touching on, what is the best way to restore the most healthy possible microbiome after you are exposed to antimicrobials? Yeah,
so as Caroline alluded to a little bit, I think this is where in the future something like fecal replacement may come in, that we may continue to use antibiotics, but then I think that it won't be unusual, probably again in the next X years, however that is, 10 years, that we might provide a stool sample, they'll freeze it, you take your antibiotics, and then they'll reconstitute it afterwards, so you'll recover from, and then the downstream impact of then you receiving someone else's microbiome, or having a long-term effect from you taking antibiotics by replacing your microbiome will probably be something that's normal. How do we do that now? Again, there is no real way to accelerate this apart from making sure that you have a healthy diet, and again, probably eating lots of dairy products, yogurts,
fibrous foods,
fermented foods, yeah, which have a greater kind of composition of microorganisms that we know can colonize our guts.
Yeah, I mean, there's been some clinical trials and various research projects that different academic institutions, at least in the US that I'm familiar with, have done, where they've taken someone's own stool, and then after someone has a stem cell transplant or are exposed to antibiotics, then they give them their own, what they call an autologous fecal transplant, and repopulate their microbiome with what they had before. But that's all only ever been done under very monitored clinical trials. And
I feel like I gave probiotics a bit of a hard time too. It's not the fact that I don't say to use probiotics. I think probiotics are fine, but I think we don't really know what their health benefits are. They won't do you any harm as far as we know. And I think that probably taking a variety of different probiotics and reading on the label what's in them is probably quite good as well. Yeah. Bifidobacterium, we think it's probably quite good. So check your probiotics for bifidobacterium.
Yeah, and lactobacillus.
And lactobacillus.
Yeah, from breast milk. Yeah, I'm not sure. I think we're done.
How long we got now? 1 minute.
One minute. One final question. Okay.
Who's a lucky person that's going to have a final question?
Oh my goodness, there's a lot about pH. Okay, is there any claim in the truth of the good effects of probiotics?
Yeah, I just alluded to that. So no, this is a bit of a kind of not a particularly strong area for science. So there are some limited studies that have been done on large cohorts, and to be honest, I don't know the literature inside out. But in my knowledge, there's nothing particularly convincing to show any real kind of major medical benefit. Not to say that there isn't. I just don't think they've been particularly well proven. Myself, we did a large randomized control trial of children taking lactobacillus and placebo, and it was double blind, which means that they didn't know what they were getting, the doctor didn't know what they were giving them, and actually to recover from diarrhea, and actually it showed absolutely no effect. So for that it didn't work, and that particular probiotic didn't work. That doesn't mean it won't work. And I think it's an area that we probably need to expand more research into to understand how we can get them to work and what organisms we need to use.
And as we've talked about, there's so many different species of bacteria that we still haven't characterized and identified, and those might be the golden ticket to some of these.
This is your job out there, right? You've got to go and get on with this. There's so many different organisms we can work on and try and turn them into probiotics to do something.
Yes.
Okay.
We'll It's been a real pleasure.
Yeah, so thank you for listening to us, and we hopefully that was useful and informative. We're based at the University of Cambridge, and we're on the website. And actually, if you have any burning questions we didn't answer, then I'm sure you can find our contact somewhere and get in touch with us. All right.
Thank you.
Thank you very much.